Hello everyone, this is Medical Gateway, and today we're going to start a new chapter, which is chapter number 33 of Guyton, Red Blood Cells, Anemia and Polycythemia. Okay, so in the first part of this chapter, we're going to discuss basically the components of blood. So blood is basically blood cells and plasma. So 45% of blood is blood cells, whereas 55% of blood is plasma. So blood cells include three types of blood cells. One is red blood cells, which are known as RBCs. Then we have white blood cells, known as WBCs. And then we have platelets. So 45% of blood is blood cells, and there are three types of blood cells: RBCs, red blood cells, white blood cells, and platelets. Then we have plasma. So 90% of plasma is liquid, which is basically water, and 10% of the plasma is solids. So the 90% of the plasma is basically liquid, which is water, and then 10% which are solids. These are important. These includes basically the most important among them is proteins. Then plasma has nutrients and different gases and ions and waste products. Okay, so blood is carrying all of these things. Blood is carrying the blood cells. Blood is carrying a lot of water in it. Then blood is carrying a lot of proteins and nutrients and gases and ions and waste products. So if we go into the detail, the most important thing in the plasma are the plasma proteins. So in the proteins, we have coagulation factors, which are basically proteins. They play their role in coagulating the blood. Then we have antibody proteins. Antibodies play their role in immunity. These both things will be discussed in detail in their respective chapters. Then we have the famous albumin, the plasma protein, and then we have fibrinogen. Fibrinogen is also involved in blood clotting. Okay. So now there's one interesting thing, that is serum. What exactly is serum? Serum is basically plasma, but plasma without fibrinogen. So serum is basically plasma without fibrinogen. Or serum is basically blood without blood cells and fibrinogen. So what is plasma? Plasma is blood other than the blood cells. So what is plasma? Plasma is blood other than the blood cells. So if we just redefine serum, so serum is basically blood other than blood cells and fibrinogen. Okay, so if we remove blood cells and fibrinogen from the blood, what is left behind is known as serum. Or if we remove fibrinogen from plasma, what is left behind is serum. So what is its significance? Okay, now whenever the blood clots, okay, you have an injury and the blood clots. So whenever the blood clots, the blood clot involves the blood cells, and along with the blood cells, fibrinogen is also trapped in the blood clot or used in the blood clot. So fibrinogen, whenever the blood clots, the blood cells are coagulated there, and the fibrinogen is also involved in the blood clot. So the blood, that blood other than the blood clot, will be the serum. Okay, so this is serum. Serum is plasma without fibrinogen. Okay, so now these were the components of the blood. Now if we discuss what are the functions of the blood, so the function of the blood can be discussed by discussing the function of each of its component. For instance, what's the function of red blood cells? Red blood cells have hemoglobin in it. The hemoglobin is the main protein which will carry oxygen from the lungs to all the tissues. Then we have WBCs, white blood cells. White blood cells have their role in the immunity. White blood cells protect our body from the foreign invaders. Then we have platelets. Platelets, as discussed before, are basically involved in preventing in clotting of the blood. And then we have proteins. There are different types of proteins, as discussed before. Coagulation proteins involved in coagulation. Antibody proteins are have their function, the immunity. Then we have albumin. Then we have fibrinogen, which is involved in the clotting. Then there are nutrients. So blood also supplies nutrients to all the organs. Then we have gases. So blood supplies oxygen to the tissues and will remove carbon dioxide from the tissues. Then ions are present in the blood. So blood is involved in ionic balance of the body. And then there are waste products in the blood. So blood is involved in removal of the waste products. So these all are the functions of the blood. So let's discuss them. So first is the nutritive functions. Then we have respiratory function. The respiratory function is related to the gases, the oxygen. Whenever we breathe, we inhale oxygen in. That oxygen is carried to all the body cells by blood. Then blood has excretory functions. The waste products of all the cells and the tissues are carried away by blood, and they are carried to the kidney, and the kidney excretes them out. Then the blood has transport functions regarding the hormones and enzymes. The blood transports hormones and enzymes from one part of the body to the other part of the body. Then blood is involved in the regulation of water balance, as plasma is 90% water, and it also has electrolytes in it. So blood is basically involved in the regulation of water balance along with the kidney. Then regulation of acid-base balance in the body. So blood has buffers in it, buffers in the form of proteins, and especially red blood cells have hemoglobin, which is a protein. So these proteins in the blood act as acid-base buffers. So that's how the blood plays its part in regulation of acid-base balance. Then regulation of body temperature. So if the body temperature rises from the normal body temperature, then the blood supply of the skin will be increased to just remove an extra amount of the heat from the body. So the regulation of body temperature is also a function of blood. Then the blood has storage function. A lot of the proteins are stored in the blood, and as the nutrients are being traveling from one part to the other, they also are being stored in the blood. Then the blood has defensive functions, as WBCs, the main defensive cells of our body, the WBCs, are a part of blood. So blood also has defensive function. So this is the start of this chapter, and these are the blood components and functions of blood. See you in next part. Hello everyone, this is the Medical Gateway, and in this part we're going to study regarding red blood cells. We're going to discuss the functions of red blood cells, but before that we'll discuss their numbers and their structure. So red blood cells, in males the total number of red blood cells are 5.3 million per millimeter square, whereas in females the number of the red blood cells are 4.7 million per millimeter square. So number of the RBCs are in a relatively greater quantity in males as compared to females. Now let's discuss its structure. So the structure of the RBC is basically in the form of a biconcave disc. So if you focus on its shape, the shape of the RBC, its diameter is around 7.8 micrometers, whereas its width on the sides, on the borders, is 2.5 micrometers, whereas its width in the center is 1 micrometer. So this difference of width in the center and the peripheries make RBCs a biconcave disc. Now, what is the significance of this structure? Basically, the amount of the membrane of the RBCs is relatively in a greater quantity as compared to the material inside the RBCs. So the material inside the RBCs is less, whereas the membrane outside the RBCs is in greater quantity. So Now, what is its significance that having more membrane as compared to the contents inside? This helps RBCs to easily squeeze through any place. If there is a blood vessel or there is a tight junction of blood vessels, RBCs can easily squeeze themselves through them. So this is a biconcave disc. This is how RBC is. What is the shape? This is the shape of the RBC. So now if the RBC has to pass through a blood vessel which has lesser diameter than 7.8 micrometers, then the RBCs will squeeze itself. Now, if this membrane, for instance, in some conditions which we'll discuss afterwards, if the amount of the membrane is reduced, then what will happen? The RBC will not be able to maintain its biconcave disc shape. The RBC will be converted into a spherical shape, and then the contents inside and the membrane both will be in proportion to each other. But what will happen in that case of the spherical RBCs rather than the biconcave RBC disc is that those RBCs will be unable to squeeze through the tight pores, and then the RBCs will be ruptured in that case, as in the case of hereditary spherocytosis, which is a hereditary disorder of RBCs, where the RBCs are converted into a spherical shape because of the less amount of the membrane outside the RBCs. So now let's discuss the functions of RBCs. So RBC has three broad functions. One is oxygen transport, the other one is carbon dioxide transport, and the third one is acid-base buffers. So let's discuss its most important function, which is oxygen transport. So we have in red blood cells, we have a protein, the oxygen carrying protein known as hemoglobin. So hemoglobin is basically an oxygen binding protein which is present inside the red blood cells. So now let's discuss exactly how it carries the oxygen and what is the amount of the oxygen carried in the RBCs by the hemoglobin. For this, we have to calculate the value of the amount of the oxygen carried by the RBCs. For that, first of all, we'll discuss that in 100 ml of blood, there are 45 ml of RBCs. And how we have calculated this, we know that 45% of the blood is basically the cells, and in the cells the RBCs are the predominant cells. So 100 ml of the blood is basically has 45 ml of RBCs. And we know that 100 ml of RBCs have 34 grams of hemoglobin. So what is the amount of hemoglobin present in RBCs? So 100 ml of RBCs have 34 grams of hemoglobin. Okay, basically we are going to calculate that what is the amount of oxygen which 100 ml of blood can carry. For that, we first have to know that what is the amount of RBCs in 100 ml of blood, and then we should know that what is the amount of hemoglobin in that 45 ml of RBCs or 100 ml of RBCs. So if 100 ml of RBCs have 34 grams of hemoglobin, then 45 ml of RBCs would have around 15 grams of hemoglobin. Okay, if 100 ml of RBCs have 34 grams of hemoglobin, then what is the amount of hemoglobin in 45 ml of RBCs? We basically want to know that what is the amount of oxygen carried by 100 ml of blood. So for that, first we have calculated the amount of the RBCs present in them. Then we will calculate the amount of hemoglobin present in these RBCs, and then we will calculate the amount of oxygen carried by that amount of hemoglobin. So if 100 ml of RBCs have 34 grams of hemoglobin, then 45 ml of RBCs will have 15 grams of hemoglobin in them. So 100 ml of blood will have 15 grams of hemoglobin. Okay, so in males, what is the concentration of hemoglobin? It is 15 grams per 100 ml of blood in males, and in females it is a little less than 15 grams. Okay, now this hemoglobin has to carry oxygen. So 1 gram of hemoglobin has the ability to carry 1.34 ml of oxygen. So 1 gram of hemoglobin has the ability to carry 1.34 ml of oxygen. So 15 grams of hemoglobin will carry 1.34 multiplied by 15 grams of hemoglobin. So 15 grams of 1 gram of hemoglobin can carry 1.34 ml of oxygen. Then 15 gram of hemoglobin can carry 20 ml of oxygen. If we multiply 15 with 1.34, the answer is around 20. So 15 grams of hemoglobin can carry 20 ml of oxygen. So now we know that 100 ml of blood has 15 grams of hemoglobin, and 15 grams of hemoglobin can carry 20 ml of oxygen. It means that 100 ml of blood can carry 20 ml of oxygen. So this is where everything was leading. Okay, so calculation requires 100 ml of blood requires the amount of the RBCs, then the amount of the hemoglobin in those RBCs, and then the ability of the hemoglobin to carry the oxygen. So finally 100 ml of blood is able to carry 20 ml of oxygen. Okay, now coming to the next function, which is carbon dioxide transport. Now as the blood can carry oxygen, which we have discussed in detail, the blood also carries carbon dioxide from the tissues to the lungs, from where the carbon dioxide is exhaled out. Now what is happening inside the red blood cells is that water reacts with the carbon dioxide, converting it into carbonic acid, H2CO3, carbonic acid. So now the carbon dioxide will travel in the blood in the form of carbonic acid. Now this is happening in the tissues. Now this carbonic acid, when this reaches the lungs, this carbonic acid is converted back into water and carbon dioxide, and this carbon dioxide will be exhaled out. This is how the blood is transporting carbon dioxide. We'll discuss in detail regarding the transport of oxygen and carbon dioxide in the chapters which will come after a few chapters. Now, which enzyme is converting this, catalyzing these reactions? So the enzyme is carbonic anhydrase. So carbonic anhydrase is the enzyme which converts water and carbon dioxide into carbonic acid, and then back carbonic acid into water and carbon dioxide. Okay, so this is how the carbon dioxide is transported. Then the blood has acid-base buffer system. So basically the proteins present in the blood can act as buffers for the acid-base regulation. So among the proteins, the hemoglobin, which is an oxygen binding protein, the hemoglobin plays the greatest amount of role in buffering of acid base. Okay, so this is also a function of blood. So these are the three main functions of blood: oxygen transport, carbon dioxide transport, and acid base buffers. So this is it. See you in next part. Hello everyone, this is Medical Gateway, and in this part we're going to study regarding the stages of erythropoiesis. So let's start it. So from where the red blood cells are produced, or exactly where the erythropoiesis takes place. So you all know the answer, that the red blood cells are formed in the bone marrow. Okay, but this is not the case in the gestational life. So in early weeks of gestation, the red blood cells are produced from the yolk sac, whereas in the middle trimester, the red blood cells are produced from liver, spleen, and lymph nodes. And then in the last months of gestation, the red blood cell production starts from bone marrow of all the bones. Okay, so RBC production from the bone marrow of all bones basically begins in the last months of gestation. In early weeks of gestation, the production is from yolk cell. In middle trimester, the production is from liver, spleen, and lymph nodes. When the last months of gestation, the production starts from bone marrow of all bones. Okay, now. So, up till when the production continues in the bone marrow of all bones? So, from last few months of gestation till the baby is 5 years old, the production of RBCs is from bone marrow of all bones. Okay, from 5 years till 20 years of age, the production of RBCs is from not the bone marrow of all bones, but the bone marrow of membranous bones and proximal portion of humeri and tibia. So from the age of 5 years till the age of 20 years, the RBCs are produced from the bone marrow of membranous bones and from the proximal portion of humeri and tibia. Okay, and after the age of 20 years, the RBC production is done by the bone marrow of membranous bones only. Okay, so when the person is of the age greater than 20 years till death, the RBCs are not produced from bone marrow of all the bones. RBCs are only produced from bone marrow of the membranous bones. So which bones are exactly the membranous bones in body? The bones which are producing the RBCs after the age of 20, basically after the age of 5, are the bones involved in the rib cage. The ribs. Now, ribs are attached behind to the vertebra, and the ribs are attached in front of the sternum. So it's the bones involved in the rib cage are involved in the RBC production, are involved in the erythropoiesis after the age of 5, and basically solely after the age of 20 years. Okay, so I'm going to just quickly revise this because it's very important. This is at times this is asked in the MCQs. So in the early weeks of gestation, RBC is produced by yolk cell. In middle trimester, RBCs are produced by liver, spleen, and lymph nodes. From the last months of gestation till 5 years of age, RBCs are produced by bone marrow of all bones. From the 5 years till 20 years, the RBCs are produced by membranous bones as well as the proximal portions of humeri and tibia. And after the age of 20, they are produced only by the membranous bones. Okay. So now, what exactly are the pluripotent hematopoietic stem cells? So all the blood cells, no, we're not just talking about the red blood cells, we're talking about all the blood cells, which include the white blood cells, the platelets, and also the lymphocytes, which are included basically in the white blood cells. So the platelets, the WBCs, the RBCs, all are formed basically from the pluripotent hematopoietic stem cells. Now, pluripotent hematopoietic stem cells are the cells which can be converted, which are finally converted into the colony-forming unit spleen, and then colony-forming unit spleen will be converted into the respective colony-forming units of the platelets, WBCs, and RBCs. So, pluripotent hematopoietic stem cells are converted into colony-forming unit spleen, and then colony-forming unit spleen will be differentiated into further 3 types of colony-forming units. One colony-forming unit will give rise to platelets. The other colony-forming unit will give rise to WBCs, and the third colony-forming unit will give rise to red blood cells. The colony-forming unit, which is giving rise to red blood cells, is known as colony-forming unit erythrocytes. And the colony-forming unit, which is giving rise to white blood cells, are known as colony-forming unit granulocytes and monocytes. Now, what is this granulocyte and monocyte? These are the types of white blood cells. So there are 2 types of white blood cells. One having granules are known as granulocytes, whereas the other ones are agranulocytes. So from there the name came, the colony-forming unit granulocytes and monocytes. Monocytes are basically agranulocytes. So this is the name. And the colony-forming unit, which will give rise to platelets, are known as colony-forming unit megakaryocytes. Okay, so these three will be formed from colony-forming unit spleen. Just there is one additional point, that before colony-forming unit spleen, there is also a stage which is colony-forming unit blast. So colony-forming unit spleen will give rise to colony-forming unit blast, and colony-forming unit blast will give further rise to colony-forming unit erythrocytes, and then colony-forming unit erythrocytes are giving rise to our red blood cells. Now, basically, what is happening, that behind we have pluripotent hematopoietic stem cells. Now, these cells are growing and multiplying, and basically also these cells are differentiating into different types. Okay, so there are two things which are happening. One is growth of the cells, and the other one is differentiation of the cells. So for the growth and the differentiation, we need some supportive substances. So for growth, we have growth inducers, and for differentiation of cells, we have differentiation inducers. Okay, so growth inducers, we have the most important growth inducer is interleukin 3. So interleukin 3 is a growth inducers. Okay, cell is growing as well as the cell is differentiating. The cells are differentiating. So we have growth inducers and differentiation inducers. Okay, so the most important growth inducer is interleukin 3. Okay, so this is the whole story. Now, I've just left one thing. In the WBCs, we also have lymphocytes. Okay, but for the lymphocytes, we have a separate lineage. The pluripotent hematopoietic stem cells are converted into lymphoid stem cells, and these lymphoid stem cells give rise to lymphocytes. Okay, so this is how the pluripotent hematopoietic stem cells are giving rise to each and every kind of blood cell. Okay, now let's move to a main topic, the stages of erythropoiesis. Okay, now the stages of erythropoiesis means the stages by which finally a mature red blood cell, which is known as erythrocyte, is formed. Okay, so mature red blood cell is known as erythrocyte. So these are the different stages of formation of a mature red blood cell, the erythrocyte. So the stages are, the first stage is, the most immature stage is proerythroblast. Then we have basophil erythroblast. Then we have polychromatophil erythroblast. Then we have orthochromatic erythroblast. Each of this stage has the name erythroblast in it. One is pro, then we have basophil, then we have polychromatophil, then we have orthochromatic. Okay, so these all are erythroblast. Then erythroblast will be converted into, these orthochromatic erythroblasts will be converted into reticulocyte, and then reticulocytes will be converted into the mature RBCs, erythrocytes. All these stages are taking place in the bone marrow till the orthochromatic erythroblast. Then in the bone marrow, the reticulocytes are formed, and then the reticulocytes finally enter from the bone marrow into the circulation, and in the circulation, the reticulocytes, which are also the immature cells, are converted into finally the mature red blood cells. Okay, so these are the stages. Okay, now if we look at the characteristics of a mature red blood cell, the erythrocyte, then we'll come to know that the mature red blood cell have hemoglobin in it. 1 point. Second one, the mature erythrocyte, the mature RBC, does not have nucleus in it. The nucleus is absent in the mature RBC. And the third point is the mature RBC also doesn't have mitochondria and ribosomes. So you have to remember 3 points. Mature RBC has hemoglobin, but it does not have nucleus, mitochondria, and RBCs. If we look at any cell, the cells have nucleus, the cells have mitochondria and RBCs, but any cell the cells doesn't have hemoglobin. So when we have to start our story from erythroblasts, proerythroblasts, the proerythroblasts have nucleus. It doesn't have hemoglobin, and it does have the mitochondria and ribosomes. Okay, and as the stages are being matured, these immature RBCs will get rid of the nucleus. The hemoglobin will start appearing, and it will get rid of the mitochondria and the RBCs. Okay, so in the stages we have to focus on 3 to 4 things. These stages are very easy. We have to focus that in which stage, whether the nucleus is present, whether hemoglobin is present, or whether mitochondria or RBCs are present or absent. Okay, so finally in the mature form, the nucleus will be absent and the mitochondria and RBCs will be absent and the hemoglobin will be present. But in the first stage, the condition is reverse. Nucleus is present, mitochondria and ribosomes are present, but the hemoglobin is absent. So this is proerythroblast. Okay, nucleus is present, nucleoli is present, hemoglobin is absent, and mitochondria and RBCs are also present. Then we have basophil erythroblasts. In this stage, nucleus is present, but the nucleoli is absent. Now, the story is moving towards the disappearance of nucleus and the appearance of hemoglobin. Okay, now, it will be easier for you to remember if you focus on the words. Basophil, orthochromatic, polychromatophil. Now, basophil. The term basophil comes from the basophilic staining. Now, we have eosinophilic staining and the basophilic staining. The eosinophilic staining was pinkish in color, whereas basophilic staining was purplish or bluish in color. Okay? Now, if you remember which part of the cell gets stained by a basophilic stain. So the nucleus is always stained by a basophilic stain. That's why I have labeled all the stages where the nucleus is present as blue, and all the stages where the hemoglobin is present as pink. Okay, so in basophilic, the basophil erythroblast is known as basophilic erythroblast because it is stained with a basophilic dye, because it has nucleus in it. Okay, so but now as we move towards the next stage, the polychromatophil erythroblast, now the hemoglobin starts to appear. The nucleus hasn't disappeared yet, but the hemoglobin has appeared. Now this erythroblast is having nucleus and also is having the hemoglobin, because hemoglobin has appeared here. So now this erythroblast will be stained by a basophilic stain as well as an eosinophilic stain. So basophilic stain will give a bluish purplish discoloration of the nucleus, whereas eosinophilic stain will be because of the hemoglobin getting stained by eosinophil. So as this stage is stained by more than one color, then because of it, its name is polychromatophil. Chromatophil is from the term chrome, and chrome is from colors. So it's polycolor, the stage having polycolors. The stage which is stained by polycolors is known as polychromatophil erythroblast, because nucleus is stained by basophil in this, and hemoglobin is stained by eosinophil in this. And in this stage, both the nucleus and the hemoglobin are present. Okay? Then we have orthochromatic erythroblast. Now, in this stage, the nucleus is lost. The nucleus is lost. Now the nucleus is not present and only the hemoglobin is present. So this stage is known as orthochromatic erythroblast. That's why I've labeled it pink, and here it's also pink. So the point from where the nucleus is absent, the bluish highlighting is absent. Okay. So basophil has only hemoglobin. Orthochromatic stage has only the basophilic stage has only nucleus. The orthochromatic stage has only hemoglobin, whereas the polychromatophil stage has both the nucleus and the hemoglobin. Okay? So nucleus has disappeared from the orthochromatic stage, whereas hemoglobin has appeared from the polychromatic stage. Okay, so in some parts, even the Guyton, it is said that in the proerythroblast stage there is little hemoglobin present, but that hemoglobin is not stained, but the hemoglobin is properly appears and is stained from the polychromatophil stage. Okay, so I've just labeled that from the polychromatic stage the hemoglobin appears. Before that, in some books, at some points, even the Guyton, it is written that hemoglobin does appear to some extent, but it is not being stained. Okay, so the nucleus has disappeared. The nucleoli disappears in the first stage, or is only present in the first stage, and disappeared in the second stage, the basophil erythroblast. Whereas the nucleus has disappears in the orthochromatic stage. Okay. And then we have hemoglobin present in all the stages, and nucleus is absent in all the stages. Okay. Now, how exactly the nucleus is lost from the cell? The nucleus is basically destroyed or basically degenerated by three mechanisms: by pyknotic degeneration, by karyolysis, and by karyoresis. Now, what exactly is pyknotic degeneration? Pyknotic degeneration means the nucleus shrinks and the nucleus goes from condensation. So shrinkage and condensation of the nucleus is known as pyknotic degeneration. Whereas there is lysis of the nucleus because of the swelling. So karyolysis involves the swelling of the nucleus, whereas karyoresis involves the degeneration of the chromatin. Okay, so if someone asks that how the nucleus is lost, you will say it by three mechanisms: the pyknotic degeneration, the karyolysis, and karyoresis. Pyknotic degeneration involves the shrinkage and condensation, karyolysis involves the swelling, and karyoresis involves the degeneration of the chromatin. Okay, so finally we have the orthochromatic stage where there is no nucleus, no nucleoli, we have hemoglobin. Okay, then there comes the retic stage, the reticulocyte stage. Okay, now the reticulocytes are present in the bone marrow. So when the reticulocytes are produced, the reticulocytes stay in the bone marrow for 2 to 3 days, and after 2 to 3 days these reticulocytes will move from bone marrow into the circulation. Okay, and there the reticulocyte in the circulation stay for 24 hours, and after that the reticulocytes are converted into the mature erythrocytes, the mature RBCs. Okay, so reticulocytes are present in the bone marrow, they stay there for 2-3 days, and then they will move to the circulation where they will stay for 24 hours in the reticulocyte form, and then they will be converted into the mature erythrocytes. Now, Now, what exactly is happening in the reticulocyte stage? When reticulocytes are present inside the bone marrow, they do have mitochondria and ribosomes. But when the reticulocytes enter into the circulation, the reticulocytes lose their mitochondria and the ribosomes. Okay? So when they lose their mitochondria and the ribosomes, then all the process is complete and they finally are converted into the erythrocytes. Now, the mature erythrocytes have no nucleus, have no nucleoli, they have hemoglobin present in them, but they don't have any mitochondria and ribosomes. Okay? So these are the stages of the erythropoiesis. One interesting point, other than the nucleus, the hemoglobin, and the mitochondria and the ribosomes, are the size of the immature and the mature RBCs. So as the RBCs are matured, their size is reduced. So the first stage, the most immature stage, the proerythroblast, the size is 14 to 19 micrometer. Then the basophil erythroblasts are smaller as compared to proerythroblast. Their size is 12 to 17 micrometer. Then the size decreases to 12 to 15 micrometers, then decreases to 10 to 15 micrometers, then 7 to 10, and finally 6 to 7.5 micrometers. Just imagine the size of 19 micrometers and imagine the size of 6 to 7 micrometers, the mature RBCs. So mature RBCs are way smaller as compared to the immature RBCs. Okay, so the most easiest way to remember the RBCs are first of all by the names of the basophil, polychromatophil, and orthochromatic. Okay, and then the immature reticulocytes, and which will enter into the circulation, which finally will be converted into the mature erythrocytes. Okay, so these were stages of erythropoiesis, a very important topic. See you in next part. Hello everyone, this is Medical Gateway, and this chapter we're going to study regarding the erythropoietin and RBC production. So what is erythropoietin? Erythropoietin is a substance, it's a hormone which basically results into RBC production from the bone marrow. And what is the main stimulus for the erythropoietin production? The main stimulus for the erythropoietin production is the hypoxia. Now, erythropoietin has to produce RBCs, and the main function of the RBCs is to carry oxygen to the tissues. So if the oxygen is not being supplied in the adequate quantity to the tissues, which is basically hypoxia, then the hypoxia will act as the main stimulus for the increased production of RBCs by stimulating the production of erythropoietin. So whenever there is hypoxia, erythropoietin is going to be produced, and erythropoietin will increase the production of RBCs. So what are the conditions which can lead to hypoxia? So these are the conditions which can be like low blood volume. Because of low blood volume, there will be hypoxia in the tissues, or anemia, or decreased in hemoglobin concentration, or decreased blood flow to the tissues, or any lung disease. All these conditions can lead to hypoxia. Lung disease can cause hypoxia because of decreased oxygenation of the tissues. Decreased blood flow can cause hypoxia because of the decreased supply to the tissues. Decreased hemoglobin will cause hypoxia because of the decreased ability to carry oxygen. And same as the anemia. And the low blood volume itself means the low blood pressure and low perfusion to the tissues and low supply of oxygen to the tissues, which is hypoxia. Now, what hypoxia is going to do? Now, if there is hypoxia, then renal tissues will also face that hypoxia. Now, whenever the renal tissues are hypoxic, they will produce hypoxia-inducible factor. And that hypoxia-inducible factor will go and will be attached on erythropoietin gene. And to be specific, hypoxia-inducible factor is attached to hypoxia response element on erythropoietin gene. So this is how it looks like. This is the DNA. For instance, this red colored is the erythropoietin gene, and the erythropoietin gene has this green colored hypoxia response element. So hypoxia-inducible factor is going to get attached to this hypoxia response element on erythropoietin gene. Now, on the response to attachment of hypoxia-inducible factor on hypoxia response element, erythropoietin genes are basically going to increase the production of erythropoietin. Now the question comes in mind, from where exactly the erythropoietin is being produced. The erythropoietin is produced from two places, the kidneys and the liver. 90% of the erythropoietin is produced by kidneys, and 10% of the erythropoietin is produced by liver. Now, it is often asked that which cells in the kidneys are producing erythropoietin. The renal epithelial cells, and to be precise, the fibroblast-like peritubular interstitial cells. So fibroblast-like peritubular interstitial cells will produce the erythropoietin. Now, what this erythropoietin is going to do? This erythropoietin will then act on bone marrow, and by acting on the bone marrow, this will lead to an increased production of the proerythroblasts, the first stage of the erythropoiesis, and the increased conversion of these erythroblasts into the mature red blood cells, and finally the increased release of the erythrocytes. Now, there are many steps in the erythropoiesis, from the proerythroblasts to finally the erythrocytes in the blood. So bone marrow, the erythropoietin is going to facilitate all of these steps. Okay, so all of these steps are facilitated by erythropoietin. So one stimulus is this hypoxia, which acts on the kidneys, and then kidneys will produce hypoxia-inducible factor, and then erythropoietin gene will result into increased production of erythropoietin, and finally the erythrocytes are produced. Now, how many days will it take from the exposure to hypoxia and the final appearance of mature erythrocytes in the blood? The whole process takes place 5 days. Okay, now after the hypoxia, immediately within few minutes, hypoxia-inducible factors are produced and erythropoietin gene gives the response. But the other steps of maturation will take some time, and after 5 days of hypoxia, the mature erythrocytes appear in the blood. So is this the only mechanism for the production of the RBCs by erythropoietin? So our hypoxia isn't the only stimulus. There is another pathway, that is the epinephrine, norepinephrine, and prostaglandins will also result into increased production of erythropoietin. So these also the sympathetic nervous system, that relate to sympathetic nervous system, the neurotransmitters, the epinephrine, norepinephrine, and the prostaglandins can also directly increase the production of erythropoietin other than the hypoxic pathway. Okay, so this is the erythropoietin, how exactly the erythropoietin is produced. Now let's discuss the factors necessary for the erythropoietin. So there are 3 types of factor. One is the general factor, then we have maturation factors, and then we have factors for hemoglobin synthesis. So general factors necessary for the erythropoiesis are that for erythropoiesis, we need erythropoietin. Obviously, we need the erythropoietin. Then we need thyroid hormone. Thyroid hormone is extremely important for erythropoiesis. We'll discuss thyroid hormone in detail when we'll discuss the endocrinology. Then we have growth factors, as discussed in the colony-forming units and pluripotent hematopoietic stem cells differentiation. We have discussed the growth factors, and we have discussed interleukin 3 there, which is causing the growth. Now, there are also a few other interleukins which are important growth factors, like interleukin 6 and interleukin 11. Then vitamins are also necessary for the erythropoiesis, like vitamin B, C, D, and E. Okay? These are the general factors. Then we have some maturation factors which will help mature the stages of the RBCs. Now, the maturation factors include vitamin B12, folic acid, and intrinsic factor. And these are extremely high yield with reference to the clinical application of this topic with reference to the anemias. For instance, vitamin D deficiency or folic acid deficiency or intrinsic factor deficiency, these all will result into maturation defects which will lead to anemia. Okay? So anemia is the decrease in the number of the RBCs. Okay. Then we have the factors for the hemoglobin synthesis. The factors for the hemoglobin synthesis include the proteins, the amino acids, the iron, copper, cobalt, nickel, and then again the vitamins. So iron, copper, cobalt, and nickel, the minerals, and the proteins and the amino acids, and the vitamins. Which vitamins are specifically relatively more relevant with reference to the hemoglobin synthesis are vitamin C, vitamin B2, vitamin B3, and vitamin B6. Okay, so this is we're done with this topic of erythropoietin and the factors necessary for erythropoiesis. See you in next part. Hello everyone, this is Medical Gateway, and today we are going to study the vitamin B12, folic acid, and their relationship with the RBC maturation. So what is the role of vitamin B12 and folic acid in RBC maturation, and what exactly is pernicious anemia? This is our topic today. So let's start with the vitamin D, exactly from where the vitamin D comes. So vitamin D is present in beef, liver, and chicken. So whenever we eat beef, liver, or chicken, the vitamin B12 B12 goes into our stomach. And when the vitamin B12 reaches the stomach, there is release of intrinsic factor from the parietal cells in the stomach. So whenever we eat vitamin B12 or whenever we eat beef, liver, or chicken, the vitamin B12 goes into our stomach, and on the entry of vitamin B12 into the stomach, the parietal cells of the stomach release intrinsic factor. So now this intrinsic factor basically has two functions. This intrinsic factor prevents the digestion of vitamin B12, and this intrinsic factor also assists the absorption of vitamin B12. So this main function of the intrinsic factor is the protection of vitamin B12 from digestion and also the absorption of vitamin B12. So intrinsic factor protects vitamin B12 and then takes this vitamin B12 and get it absorbed into the mucosal cells of the ileum. So when the vitamin B12 is absorbed into the mucosal cells of ileum, then the vitamin B12 moves into the blood via pinocytosis. So finally, the vitamin B12 we have eaten goes into the blood. So from blood, the vitamin B12 goes into the liver, and in the liver, the vitamin B12 is stored. And this vitamin B12 will be used by the bone marrow whenever the maturation of RBC is needed. So the main function of the vitamin B12 with reference to the erythropoiesis is that vitamin B12 helps in maturation of red blood cells. So if there is deficiency of vitamin B12, then it will result into defect in maturation of RBCs. I'm going to quickly revise this, that vitamin B12 comes from beef, liver, and chicken. Whenever it goes into the stomach, the parietal cells of the stomach release intrinsic factor. Now this intrinsic factor will prevent vitamin B12's digestion and will assist its absorption into the mucosal cells of the ileum, from where the vitamin B12 moves into the blood via pinocytosis, and then this vitamin B12 will be stored in the liver. And then this vitamin B12 will be utilized by the bone marrow whenever the maturation of the RBC is required. Okay. Now, what exactly is pernicious anemia? And how pernicious anemia is related to this vitamin B12 or intrinsic factor? So let's discuss it. So, okay. Now, in pernicious anemia, there is atrophy of gastric mucosa. Now, whenever there is atrophy of gastric mucosa, it will result into decreased gastric secretions. Now, mucosa is atrophied, so from where the secretions are going to come? So there will be decreased secretions. And when there are decreased stomach secretions, then it means there will be decreased production of intrinsic factor. And when there will be decreased production of intrinsic factor, now there is no intrinsic factor or less intrinsic factor to prevent the digestion of B12 and to assist the absorption of B12, there will be decreased vitamin B12 absorption. Okay? So this is basically pernicious anemia. And vitamin B12, whenever there is decreased vitamin B12, this will lead to maturation defect of RBCs, which is basically pernicious anemia. So anemia because of the vitamin B12 deficiency, and to be precise, anemia because of the intrinsic factor deficiency is the pernicious anemia. Okay. So once again, in pernicious anemia, there is atrophy of gastric mucosa, which results into decreased production of intrinsic factor, and which further leads to decreased vitamin B12 absorption. So in the body, if there is decreased vitamin B12, so does it mean it is only because of the pernicious anemia? No, it can also be because of some other causes like dietary deficiency. If someone is not suffering from malnutrition or not eating the things which have vitamin B12 in it, then it is a dietary deficiency of vitamin B12. And the second one is the malabsorption. If there is some malabsorptive defect, then the vitamin B12 will be deficient from the body. So vitamin B12 can be deficient because of the dietary deficiency, because of the malabsorption, or because of the intrinsic factor deficiency. Now, what will happen to the maturation of RBCs, and how exactly deficiency of vitamin B12 is going to affect maturation of RBCs? So whenever there is vitamin B12 decreased level of vitamin B12 in the body, there will be decreased formation of thymidine triphosphate. Now, if you remember the bases in the DNA, there were 4 types. One was guanine, cytosine, thymidine, and adenine. So there was adenine, guanine, cytosine, and thymidine. Now, and whereas in the RNA, in place of thymine, there was uracil. So now, what B12 does, B12 basically helps in the formation of thymidine triphosphate. So when there is B12 deficiency, there will be decreased thymidine triphosphate. And when there is decreased thymidine triphosphate, it will result into decreased DNA maturation. Okay? So vitamin B12 basically helps with the DNA maturation by formation of thymidine triphosphate. Okay, so this is how the decrease in vitamin B12 deficiency is affecting the DNA maturation. Now, DNA maturation is being affected, but there is no defect in the RNA synthesis or RNA maturation. So there is defect in the DNA maturation, but there is no defect in the RNA synthesis. Now, interestingly, where is DNA located? DNA is located in the nucleus. And where is RNA located? RNA is basically, there are 3 types of RNA: the messenger RNA, the ribosomal RNA, and the transfer RNA. So messenger RNA goes from nucleus into the cytoplasm, and then there in the cytoplasm there are ribosomal RNA and the transfer RNA. So all these RNAs are basically working in the cytoplasm. So now the nucleus has DNA and the cytoplasm has RNA. So the main component of the nucleus, the DNA, that is not being matured, whereas the synthesis of the RNA, and from the RNA, the protein synthesis inside the cytoplasm is taking place well. So what is going to happen? So the nucleus is not maturing, whereas the cytoplasm is maturing normally. So when the nucleus is not maturing and the cytoplasm is maturing normally, it means that the size of the nucleus is not growing, whereas the size of the cytoplasm is growing with time. Or the size of the nucleus is not growing at the normal pace, whereas the size of the cytoplasm is growing with the normal pace. So there will not be any sync between the relative growth of the nucleus and the relative growth of the cytoplasm. The cytoplasm will grow way more than the size of the nucleus. Now this, as there is no sync between the growth of the both parts, the nucleus and the cytoplasm, this condition is known as nucleocytoplasmic asynchrony. Okay, so this is nucleocytoplasmic asynchrony. Okay, so now one more thing is that when the cell goes for replication, whenever the size of the nucleus is reached to a maturer level. So whenever the growth of the nucleus is complete, and it's then the nucleus goes into replication. Now, what is happening is, in this case, that the nucleus is not growing, or to be precise, the DNA inside the nucleus is not maturing, and if the DNA is not maturing, then the cell replication will be delayed. So the cell replication is delayed, but the cytoplasm is continuously growing. So in this case, there will be a relatively smaller nucleus, but there will be a very large sized cell. That's why this anemia, because of the vitamin B12 deficiency, is known as macrocytic anemia. In this anemia, the size of the red blood cell, the mean corpuscular volume of the red blood cells, the MCV of the red blood cells is greater than normal. Okay, so in vitamin B12 and folic acid deficiency, both results in macrocytic anemias. Okay, because the cell grows large before the nucleus matures and replicates. Okay, so there are three things. Number one, the DNA is not being matured. Second, there is nucleocytoplasmic asynchrony. And third, the cell is growing large way before the nucleus is matured and it replicates. So that's why this whole condition is known as macrocytic, or the large sized cells, and this is anemia. So this is macrocytic anemia. Now, why this is anemia? Okay, now the point is this. Whenever there is a nucleocytoplasmic asynchrony and the cell grows way more than the normal size, then the membrane of the red blood cells will become flimsy. There will be increased fragility of the red blood cell membrane. So whenever the fragility of the membrane is increased, this RBC is easily ruptured. So this RBC will have a short life. This large size, this macrocytic RBC, this RBC having a increased mean corpuscular volume will have a short life, and because of this short life, it will result into anemia. Okay, so this is pernicious anemia. To be specific, pernicious anemia is because of the pernicious anemia is. Any vitamin B12 deficiency is not pernicious anemia. Pernicious anemia is basically the vitamin B12 deficiency because of the intrinsic factor deficiency, and that is because of the atrophy of the gastric mucosa. Okay, and then vitamin B12 deficiency due to any reason will result into this macrocytic anemia because of this thymidine triphosphate, decrease in thymidine triphosphate formation and nucleocytoplasmic asynchrony. Okay, so this is the vitamin B12, the role of vitamin B12 with reference to the RBC maturation, and this is pernicious anemia. Now let's move to the folic acid. Now folic acid is present in green vegetables, present in fruits, present in meat, liver. So their deficiency, if the folic acid, if the person is not eating them, or there's deficiency of folic acid due to any reason, then it will also lead to the same maturation failure as in the case of vitamin B12 deficiency. So that's why vitamin B12 and folic acid, these topics are always studied combined, because they both lead to maturation failures. Okay, now folic acid deficiencies aren't that much common. The reason behind is that body has huge amount of extra stores of folic acid. So if someone is having a decreased intake of folic acid in diet for some time, then it doesn't matter until unless someone is severely malnutrition because of the diet, like tea and toast diet, for a very long duration of time, then they might go into folic acid deficiency, and then the folic acid will lead to the maturation failure, just like vitamin B12. Okay, so this is a very important topic, especially with reference to the clinically speaking, because of the pernicious anemia, and that's why in diet the vitamin B12 and the folic acids are extremely important. Okay, so this is it. See you in next part. Hello everyone, this is Medical Gateway, and in this part we're going to study regarding the structure of hemoglobin and the types of hemoglobin. So let's start. So hemoglobin is a very important protein which is present basically in the red blood cells, and its function is to carry oxygen. So how exactly hemoglobin is formed? So hemoglobin chain is formed by the combination of heme and a polypeptide, which is basically globin. Okay, so it's the combination of heme and globin, or heme and polypeptide. Now, how exactly heme is formed? So heme is formed in 3 steps. Let's discuss that. So the first step is 2 succinyl CoA combined with 2 glycine in order to form a pyrrole ring. Okay, so 2 succinyl CoA combined with 2 glycine to form pyrrole, and 4 pyrroles combine to form a protoporphyrin 9 ring. 4 pyrroles combine together to form a protoporphyrin 9 ring. So this is 1 pyrrole. So this is 1, 2, 3, 4. These 4 pyrroles collectively form a protoporphyrin 9 ring. So when an iron is added to this protoporphyrin 9 ring, this results into the formation of heme. So this complete is heme. 4 pyrroles making a protoporphyrin 9 ring along with 1 iron is basically heme. So this is a pyrrole, and 4 pyrroles are collectively forming a protoporphyrin 9 ring, and protoporphyrin 9 ring along with iron forms heme. Now heme along with a globin, which is basically a protein, a polypeptide, will form hemoglobin chain. Okay, so this is one hemoglobin chain. Now the hemoglobin chains are of many types. So these are alpha, beta, delta, gamma, zeta, and epsilon. So these are the alpha, beta, delta, gamma, zeta, and epsilon. So these are different types of hemoglobin chain. Okay, now this is hemoglobin chain. Now a hemoglobin molecule basically consists of 4 hemoglobin chains. So a hemoglobin molecule looks like this, and it has 4 hemoglobin chain. So this is one hemoglobin chain, and these are the types of a single hemoglobin chain. So a hemoglobin molecule consists of 4 such hemoglobin chains. So 4 hemoglobin chains having a single iron, so 4 iron. So this is a hemoglobin molecule. For instance, this is hemoglobin A, which has 2 hemoglobin alpha and 2 hemoglobin beta. Now let's discuss in detail different types of hemoglobin molecules. Okay? So what are the types of hemoglobin? So in adults, there are 3 types of hemoglobin: hemoglobin A, hemoglobin A2, and hemoglobin F. Now the most common is hemoglobin A, which is 97%, then hemoglobin A2, which is 2%, and then hemoglobin F, which can vary in different conditions. Okay, now, so hemoglobin A. So this is the most commonly found hemoglobin inside the body. Okay, so now each if you look here, each hemoglobin molecule consists of total 4 chains. So hemoglobin A is made up of 2 alpha chains and 2 beta chains, whereas hemoglobin A2 is made up of 2 alpha chains and 2 delta chains, whereas hemoglobin F, which is fetal hemoglobin, is made up of 2 alpha chains and 2 gamma chains. Okay, so if we see what is common in all the hemoglobin molecules, that in the adult life, all of them, hemoglobin A, hemoglobin A2, and hemoglobin F, all of them have 2 alpha chains. Okay, now the difference is in the other 2 chains. So hemoglobin A has 2 beta chains, hemoglobin A2 has 2 delta chains, and hemoglobin F has 2 gamma chains. Okay, so you have to remember this, the amount of the chains, the different types of chains in different hemoglobin molecules. So I'm talking about the adult life. So if we talk about the fetal life, okay, so in embryo, there are different hemoglobin chains, different types of hemoglobin, and in fetal life, there are different types of hemoglobin. So embryo is when the unborn child is from 0 to 7 weeks, and the fetal life is 8 weeks onwards, which means 8 weeks till the birth. Okay. So in embryo, from 0 to 7 weeks, there are two types of hemoglobins. One is GOA1 and the other one is GOA2. So GOA1 type of hemoglobin has 2 zeta chains and 2 epsilon chains, whereas GOA2 has 2 alpha chains and 2 epsilon chains. Okay? So in the embryo, from 0 to 7 weeks, there are two types of hemoglobin molecules present. One is GOA1 and the other one is GOA2. There is no hemoglobin A, no hemoglobin A2 here, no hemoglobin F here. So GOA1 is made up of 2 zeta chains and 2 epsilon chains, and GOA2 is made up of 2 alpha chains and 2 epsilon chains. Now in GOA1 and GOA2, if you look here, what is the common chain? That is 2 epsilon chains are common in GOA1 and GOA2. So the difference is here in 2 zeta chains and 2 alpha chains. Okay. Now coming towards the 8 weeks onwards, fetal life. So in fetal life, we have hemoglobin F. The hemoglobin F from fetal. So this hemoglobin F and this hemoglobin F are the same. So basically, hemoglobin F present in the fetal life from 8 weeks onwards is basically 2 alpha chains and 2 gamma chains, as told earlier, 2 alpha chains and 2 gamma chains. Okay, so it's very easy to memorize because in the adult life, all of them have 2 alpha chains. The difference is A has beta chains, A2 has delta chains, and fetal have gamma chains. Okay, then GOA1 and GOA2 both have epsilon chains, 2 epsilon chains, the difference in between the zeta and the alpha chains. Okay, so this is how the hemoglobin is formed, and this is the basic structure of the hemoglobin, a hemoglobin chain, and this is how one hemoglobin molecule is made up of 4 different types of hemoglobin chains, and in the adult life we have 3 different types of hemoglobin molecules, where in the fetal life there are other 3 types. Okay, so the most important among all of them is hemoglobin A, because in adult life 97%, which is the predominant percentage, 97% is hemoglobin A, which is 2 alpha chains and 2 beta chains. If not defined, generally we're talking about hemoglobin, then it means most of the times we're talking about hemoglobin A, which is 2 alpha chains, which has 2 alpha chains and 2 beta chains. So this is it regarding the hemoglobin structure and formation and its types. See you in next part. Hello everyone, this is Medical Gateway, and today we're going to study regarding transport and storage of iron. So if we eat something which has iron in it, the iron goes into our tummy, so the iron basically reaches the intestines. Now there comes apotransferrin from liver, and iron combines with the apotransferrin in the intestine, and transferrin is formed. Now transferrin is a form basically in which the iron is being transported in the blood. Now this iron in the form of transferrin is now being transported where it is utilized. Now iron is basically utilized for erythropoiesis and hemoglobin formation. So this iron goes to the bone marrow, and where there is erythropoiesis and erythroblast formation, and it is being utilized in the formation of hemoglobin. Hemoglobin is an oxygen carrying protein in the red blood cells, and as discussed in the previous parts, hemoglobin has iron in its structure. So iron is utilized to form hemoglobin. Now, is this the only thing which is formed by iron? No, there are some other things which are also formed by iron, like myoglobin. Now, as hemoglobin is an oxygen carrying protein in the red blood cells, myoglobin is the oxygen carrying protein in the muscles. Now, Then we have cytochrome and cytochrome oxidase. Iron is also utilized to form cytochrome and cytochrome oxidase. Now, these are cytochrome and cytochrome oxidase are involved in the electron transport chain, which we have read in the mitochondria, in the chemiosmotic mechanism of ATP production in mitochondria. Then we have peroxidase and catalase. Now, these are the enzymes. Peroxidase is an enzyme which is basically involved with the oxidation of things, and whereas catalase is the enzyme which keeps a check on the process of oxidation so that it remains in a balanced form. Okay, so these are the things which are formed by iron. Now, iron is utilized in forming these all substances. Then the extra amount of the iron goes to the liver and reticular endothelial cells of bone marrow for storage. Now, this iron will be converted into the ferritin. The main storage form of iron is ferritin. So iron combines with apoferritin and it is converted into ferritin. Okay, just similar to the previously where apotransferrin was made by liver, iron combines with the apotransferrin and transferrin is formed. Similarly, now iron combines with the apoferritin and ferritin is formed. Now, transferrin is the form in which the iron is transported, whereas ferritin is the form in which the iron is stored. Now, this ferritin is soluble, small storage form. Now, if the amount of the iron exceeds the amount of the apoferritin, this means that we have additional iron. Now, this additional iron will be stored in the form of hemosiderin. Okay, now if we have excessive amount of iron in body, that we have utilized all the apoferritin stores, so ferritin has reached its maximum capacity of production. Now, this iron will be converted into another storage form, which is hemosiderin. Now, hemosiderin is pretty different from ferritin. Ferritin is a soluble form, whereas hemosiderin is insoluble form. Okay, ferritin is a small storage form, whereas hemosiderin have large clusters. It has such large clusters that it is visible from the microscope. Okay. Now, so the iron has transferred to the bone marrow, it is utilized, it is stored. Now, one other source of iron is whenever these RBCs, the red blood cells, complete their 120 days of life cycle, these RBCs are destroyed. These RBCs are basically eaten up by monocyte macrophage system, and these RBCs are destroyed, and their iron just comes out of the RBCs. Now, this iron goes to the storage form. This iron is being stored in the form of ferritin. Okay, so we have discussed all these things, but just one thing is left regarding the iron is that how the iron is basically excreted out of the body. So that there is daily loss of 0.6 mg of iron through feces. Okay, so there is daily loss of 0.6 mg of iron through the feces. And in the menstruating females, the daily loss of iron during menstruation is 0.7 milligrams. So during the menstruation, the combined loss of iron becomes 0.7 plus 0.6, which is 0.13 mgs. Okay, so this is all regarding the transport and storage of iron. The things to remember are what are the things which are formed by the iron, and then the most two important things are that iron is being transported in the form of transferrin, whereas it is stored in the form of ferritin. So this is all regarding transport and storage of iron. See you in next part. Hello everyone, this is Medical Gateway, and in this part we're going to study regarding the hemoglobin abnormalities. So we have two types of hemoglobin abnormalities which we will discuss in this part. One is the sickle cell anemia and the other one is thalassemia. So what happens in sickle cell anemia? So in sickle cell anemia, there is hemoglobin S. So what is hemoglobin S? Previously we have studied hemoglobin A, hemoglobin A2, hemoglobin F. Now this is hemoglobin S. This is a specialized hemoglobin which is present in the cases of sickle cell anemia. Now hemoglobin S consists of 2 alpha chains and 2 beta chains, and now you will say that this is perfectly normal, having 2 alpha chains and 2 beta chains. But just focus here. On this beta chain, there is a small s, which means that this is not the normal beta chain. This is the beta chain which is giving rise to the hemoglobin S, which will cause sickle cell anemia. Now, what is the difference between a normal beta chain and this beta chain of the sickle cell hemoglobin? Now, in this beta chain, the amino acid glutamic acid is replaced by amino acid valine. So, amino acid valine replaces the normal amino acid glutamic acid in the beta chains. And whenever this happens, this will not form the normal hemoglobin, it will form the hemoglobin S, which is the sickle cell hemoglobin. Now, how this chain and how this hemoglobin behaves differently from the normal hemoglobin. Now, what happens is that whenever this hemoglobin is exposed to low oxygen, this hemoglobin will be converted into elongated crystals inside the RBCs. Now, whenever this hemoglobin S, is exposed to low oxygen concentration, this hemoglobin will be converted into elongated crystals inside the RBCs. Now, these elongated crystals will get stuck in the capillaries. Now, these RBCs will lose their normal biconcave disc shape because of the elongated crystals present inside them. And these RBCs are prone to get stuck in the smaller capillaries. And when these get stuck in the smaller capillaries, then these RBCs will rupture. Okay, and these RBCs will rupture and are eaten by the macrophages, giving rise to decreased in the RBC count, giving rise to anemia. Okay, so what is happening? That glutamic acid is being replaced by valine, which makes this hemoglobin S to be converted into crystals on exposure to low oxygen. Now, these crystals, whenever these crystals are formed inside the RBCs, RBC lose their normal biconcave disc shape, and these RBCs are converted into a sickle shape because of the elongation of hemoglobin crystals inside the RBCs. So this is the RBC. Okay, now, and it has converted into a sickle shape. Now, what exactly is a sickle? Well, sickle is used in the gardening, so we're talking about that sickle. Okay, now, so this is the shape of the sickle. Now, RBCs are converted into this. Now, this RBC can easily get stuck in smaller capillaries, and when an RBC gets stuck in a smaller capillary, its fate is to get ruptured and got eaten by monocyte macrophage system. Okay, and when they will rupture, this will result in a decrease in the number of the RBCs, causing anemia. And because of its specific sickle shape, this condition is known as sickle cell anemia. Okay, now one interesting thing which is taking place here is that whenever the smaller capillaries get stuck by these elongated crystals of sickle-shaped RBCs, now smaller capillaries are blocked. Now, when these smaller capillaries are blocked, because of this blockage, there will be further reduction in the blood supply to the cells. And this reduction in the blood supply to the organ will cause additional low oxygen in those tissues, and this additional low oxygen will cause more sickling, more production of sickle-shaped RBCs. More production of sickle-shaped RBCs will lead to more RBCs getting stuck up in the capillaries. More capillaries are blocked, which will result into more reduced supply in the blood, in the tissues, and further reduced blood supply will cause an additional low oxygen. So this cycle will keep going on and on. The low oxygen causing sickling and the sickle-shaped cells are further decreasing the oxygenation of the tissues by blocking the blood vessels. So this is a positive vicious cycle which gets pretty bad. So this condition is known as sickle cell crisis. Now in sickle cell crisis, there will be severe anemia, there will be severe blockade of blood vessels. So this condition is also known as vaso-occlusive crisis. The vaso-occlusive, that blood vessels are being occluded by these abnormal RBCs. Now in the vaso-occlusive crisis, as the smaller capillaries are getting stuck by these RBCs, it will result into increased amounts of body aches and pains. It will result into fatigue, breathlessness. Because of the stoppage of the blood, there will be swelling and pooling of the blood, and then there might be blurred vision if the blood vessels of the eye are involved with this blockage of small capillaries. So this is sickle cell crisis. Okay, so this is sickle cell anemia. This is a very important topic. This is commonly asked in exams and MCQs. The other one is the thalassemia. So we are discussing about alpha thalassemia. We will not go in too much of detail of thalassemia. We'll just keep the topic to the details which are written in the Guyton. So now alpha thalassemia has two types of hemoglobin. One is hemoglobin H. Now from the name, it is obvious that alpha thalassemia involves the alpha chain. Now, in one case, that alpha chain is missing. Now, if the alpha chain is missing, it gets replaced by two more beta chains. So that is known as hemoglobin H. So in alpha thalassemia, hemoglobin H might appear. So when the hemoglobin and the structure of the hemoglobin H has 4 beta chains. It is not 2 alpha chains and 2 beta chains. This is alpha chains are missing, and all of the 4 chains are beta chains. And when all of the 4 chains are beta chains, this is known as hemoglobin H. Okay. Then we have hemoglobin baths. If there are no alpha chains and no beta chains, then all of the 4 chains will be 4 gamma chains. If there are 4 gamma chains, then this hemoglobin is known as hemoglobin baths. Okay, so in alpha thalassemia, there might be 2 types of hemoglobin present. One is H hemoglobin, and the other one is hemoglobin baths. So hemoglobin H has 4 beta chains, and hemoglobin baths have 4 gamma chains. Okay, so these were the hemoglobin abnormalities written in the Guyton. So this is it. See you in next part. Hello everyone, this is Medical Gateway, and in this part we are going to study regarding the lifespan of RBCs. So the lifespan of red blood cells is 120 days, which is 4 months. So after 4 months, the red blood cells basically die. So why exactly the red blood cells die after 4 months, and what is the mechanism? So in order to understand, we have to understand first of all few things. That red blood cells doesn't have nucleus, mitochondria, or endoplasmic reticulum. So if a cell is not having these things, then how the cell is going to produce energy? But still, we know that RBCs can produce ATPs. So how do they produce ATPs without having the mitochondria? So they produce energy because of the cytoplasmic enzymes present in the red blood cells. The red blood cells doesn't have nucleus, mitochondria, or endoplasmic reticulum, but they do have cytoplasmic enzymes within them. Okay, now, so what are the function of these cytoplasmic enzymes. These cytoplasmic enzymes basically maintain the cell membrane pliability of RBCs. So cell membrane pliability is very important. The cell membrane of the RBCs is in such a way that the RBCs have a biconcave disc structure, and the cell membrane is much more as compared to the content inside the RBCs. Therefore, this cell membrane and its pliability and its flexibility allow the red blood cells to squeeze through any small opening. Second one is the cytoplasmic enzymes help in maintaining the membrane transport to the cell membrane of red blood cells. Now, the membrane transport. The membrane transport is very necessary in RBCs because the oxygen has to pass the membrane and the carbon dioxide has to cross the membrane. So that's why cytoplasmic enzymes maintain this transport. Then cytoplasmic enzymes maintain ferrous form of iron. So iron has to be present or maintained in ferrous form, not the ferric form. Okay, so that is maintained by cytoplasmic enzymes. Then finally, the cytoplasmic enzymes prevent oxidation in the RBCs. So these are the four functions: maintaining cell pliability, and then maintaining the membrane transport, and then maintaining the ferrous form, not the ferric one, and then prevention of oxidation. Now, what will happen if these enzymes become less active? So after 120 days or with time, basically what is happening is that these enzymes which are performing these functions, these enzymes become less active. So when they will become less active, then they will not be able to maintain the cell membrane pliability. The membrane will become fragile. Now just imagine an RBC at 84 micrometer RBC, which has fragile membrane, is passing through a 3 micrometer splenic red pulp trabecular space. Definitely an 84 micrometer fragile membrane cannot pass through 3 micrometers space, so it will be destroyed while passing through the splenic trabecular spaces. Now, when they will be destroyed, so in the liver and the spleen, there are already present the macrophages. These macrophages will phagocytose the RBC, and specifically the hemoglobin present inside the red blood cells. Now, when the hemoglobin is phagocytized and the RBC is dead basically after 120 days, now what is going to happen with this hemoglobin when the macrophages eat them up? Now, as you know, hemoglobin is made up of heme and globin. So globin is a protein. So the globin part detaches, and heme is made up of iron and a porphyrin ring. Okay? So this iron is basically goes to the ferritin stores, or this iron goes to the formation of the new RBCs. So there are two things which can be done with this iron. Either it goes to the ferritin stores, or this iron can be utilized to make new RBCs. And what is going to happen with this porphyrin? So this porphyrin will be converted into bilirubin, and that will be released in bile. So this is that bilirubin, which when the level of this bilirubin is raised, that is known as jaundice, the yellowish discoloration of skin. So this is bilirubin. Whenever there is increased breakdown of RBCs, increased amount of porphyrin is produced and increased amount of bilirubin is produced, which gives yellowish discoloration to the skin. So this is the regarding the topic lifespan of RBCs. The RBCs have 120 days of lifespan. And why they are being dying after 120 days, because the cytoplasmic enzymes which are maintaining all these functions becomes less active and the cell is finally destroyed. Okay, so this is it. See you in next part. Hello everyone, this is Medical Gateway and we are studying chapter number 33 of Guyton, and in this part we are going to study regarding anemias. So morphologically, anemias are divided into 3 types. So what exactly is anemia? Anemia is decreased in red blood cell mass or decrease in hemoglobin concentration. So either decrease in red cell mass or decrease in high hemoglobin concentration is known as anemia. So morphologically, anemia is divided into three types. One is microcytic anemia, then we have normocytic anemia, and then we have macrocytic anemia. So the normal size of the red blood cell is 80 to 100. Okay, so MCV is 80 to 100. So all the anemias having the normal size are known as normocytic anemias. So if the size of the red blood cell is less than 80, okay, so if the MCV, mean corpuscular volume of RBC, is less than 80, then it is said to be microcytic anemia. And if the size of the RBC is greater than 100, the MCV is greater than 100, then it is known as macrocytic anemia. So what are the different types of microcytic anemia? So microcytic anemia include iron deficiency anemia, anemia of chronic disease, thalassemia, and sideroblastic anemias. Okay, when you have CBC report, there you have hemoglobin. So if the hemoglobin is lower than normal, then you go to the MCV and you see what is the MCV of the red blood cell. So now if the MCV is less than 80, then it means it is microcytic anemia, and then it means the causes are these four. Either the anemia is due to iron deficiency anemia, or due to the chronic disease, or due to thalassemia, or it is because of sideroblastic anemia. Now, iron deficiency anemia, as you all know, the hemoglobin consists of iron. If there is iron deficiency, then the proper hemoglobin cannot be formed, which results into iron deficiency anemia. Then we have anemia of chronic disease. Any chronic disease can result into anemia, and that anemia is also a microcytic anemia. Okay, so anemia of chronic disease. Then we have thalassemia. As discussed previously, thalassemia can be alpha thalassemia or beta thalassemia. In alpha thalassemia, there are some genetic defects with reference to alpha chain of hemoglobin, whereas in beta thalassemia, there is a genetic disorder with reference to the beta chains of hemoglobin, and in them the normal hemoglobin is not produced. So the anemia because of thalassemia is also a microcytic anemia. Then we have sideroblastic anemia. In sideroblastic anemia, basically there is defect of iron addition to the hemoglobin. Now what happens in here is that the iron starts to accumulate in the mitochondria, and that those mitochondria filled with iron will accumulate around the nucleus in the RBCs. So normally the RBCs does not have nucleus, but in sideroblast anemia there are blasts. So there is the RBC is nucleated, and it has iron-filled mitochondria surrounding the nucleus in the form of sideroblasts. Sorry. Then we have normocytic anemia. In the normocytic anemia, now first discuss the macrocytic anemia. In the macrocytic anemia, we have vitamin B12 deficiency, we have folic acid deficiency, then we have liver disease, and we have drugs. So we have discussed regarding the vitamin B12 and folic acid deficiency. Basically, in these deficiencies, there is defect in the maturation of the red blood cells. Okay, so when the red blood cells are not matured, there is nucleocytoplasmic asynchrony, which results into relatively larger size of the red blood cells. A smaller sized nucleus with the larger sized cytoplasm, which is giving a macrocytic picture of the red blood cells, where the mean corpuscular volume is greater than 100. Then we have liver disease. Now, in liver disease, there is also deficiency. Liver disease might cause the deficiency of vitamin B12 and folic acid. So, liver disease can also lead to macrocytic anemia. Then we have some drugs like methotrexate and many others, which can cause anemia, and the picture is macrocytic in this case. Okay, then we have normocytic anemias. The normocytic anemias can be because of production defect, and it can be because of hemolysis. So first, let's discuss the production defect. Now, if the RBCs are not being produced, for instance, in the case of aplastic anemia, now aplastic or aplasia means that the bone marrow is not producing the red blood cells. So that is known as aplastic anemia. Okay, then we have renal disease. In renal disease, the RBCs will not be produced in the normal quantity because the kidneys have to produce the erythropoietin, and erythropoietin then produces the facilitates the process of erythropoiesis. So in the case of renal disease, because of the low erythropoietin levels, there is anemia, and that anemia is normocytic in picture with the MCV 80 to 100. Then we also have malignancies. In the malignancies, there is also some production defect of RBCs from the bone marrow. Okay, now, so normocytic have one is production defect, and the other one is hemolysis. Hemolysis also causes normocytic anemias. Now, hemolysis include, number one, hereditary spherocytosis. Now, as you all know that the RBCs are normally biconcave in shape, so in hereditary spherocytosis, the RBCs are converted into a spherical shaped RBC. RBCs lose their membrane and they're converted into a spherical shaped RBC. Now this spherical shaped RBC, whenever it is passing through any small pore, this RBC will be ruptured, causing anemia. So when the RBC is ruptured, the RBC is hemolyzed. So this is hemolysis, and this will give a normocytic picture. Then we have sickle cell anemia. As we have discussed previously, the sickle cell anemia, there are sickle-shaped RBCs which are produced because of the exposure of hemoglobin S to the low oxygen. So now these previously normal hemoglobin will be converted into crystals, and those crystals will convert the RBC into a sickle cell shaped RBC. Now that sickle cell shaped RBC will get stuck in the capillaries, and then they will be hemolyzed, giving rise to anemia. Okay, then we have erythroblastosis fetalis. Now we'll discuss erythroblastosis fetalis just a few chapters after this. In erythroblastosis fetalis, there is hemolysis in baby. We'll discuss in detail afterwards. Then we have G6PD deficiency. Now, whenever there is G6PD deficiency, this also leads to anemia. Now, G6PD protects the red blood cells from the oxidative stress. Now, whenever there is G6PD deficiency, the red blood cells are more prone to hemolysis, giving rise to normocytic anemia. Then we have paroxysmal nocturnal hemoglobinuria. Now, hemoglobinuria. Urea is from the urine and hemoglobin is hemoglobin. So if hemoglobin starts appearing in the urine, that is hemoglobinuria. So nocturnal hemoglobinuria means, and basically paroxysmal nocturnal hemoglobinuria, there is hemolysis, and that hemolysis takes place at night. And when the person wakes up, so the first urination of the day gives a picture of hemoglobinuria, the hemoglobin in the urine. Then we have autoimmune hemolytic anemia. Now, in autoimmune hemolytic anemia, the hemolysis of the red blood cells is because of the autoantibodies, which are causing the RBCs to rupture. Okay. So these are briefly the morphological classification of anemias. The patient comes to you with, for instance, the patient is fatigued. You go for a CBC report, then in that you see the hemoglobin of the patient is low. Then you will immediately see the MCV of the patient. So now you have to see whether the MCV is normal, or MCV is low, or MCV is high. Then from that, and with the combination of history and few detailed tests, we can just rule out that finally the cause of the anemia. Okay, so this is morphological classification. Now let's discuss a few other points regarding the anemia. So now we have blood loss anemia. For instance, if someone suffers from a car accident and then there is rapid hemorrhage and he goes to the hospital, then what happens, how the body responds to it? The body replaces the lost blood with the fluids immediately in 1 to 3 days. If someone has lost 1 liter of blood, then in order to maintain the blood pressure, the body will replace that blood with the fluids in the next 1 to 3 days. Now, this will cause hemodilution. This will cause dilution of the RBCs in the blood. Now, this hemodilution will lead to anemia, that is relatively decreased mass of RBCs or decreased concentration of hemoglobin. Now, but this anemia will return to normal in 3 to 6 weeks. Because the body will start to produce more erythropoietin and will start to generate more RBCs to compensate the loss. So this is in the case of acute loss. Now, if there is chronic blood loss, the blood loss is continuing from one part to another. For instance, there are some GI cancers or continuously bleeding ulcers or hemorrhoids which continuously keep bleeding. In that case, the iron is not absorbed as rapidly as the blood is being lost. So now the body tries to increase the RBC production, but the speed of the loss of the blood is greater than the speed of the absorption of the iron. So what happens is that the body will keep on trying to increase the RBC production. Now, what will happen that body will start producing smaller RBCs than the normal, with less hemoglobin present in them. Okay, so now in the chronic blood loss, the body cannot compensate by the production of the RBCs. There is less iron as compared to the blood lost, but the body is pushing the bone marrow to produce more RBCs. The bone marrow will start producing RBCs, but they will be RBCs of smaller size than the normal, and they will have less hemoglobin as compared to the normal RBCs. Then we have hereditary spherocytosis. As we have discussed earlier, that RBCs lack normal loose bag-like membrane in hereditary spherocytosis. The shape becomes spherical instead of biconcave, which makes them very difficult to pass through the red splenic pulp or squeeze through the red splenic pulp, and because of that they get ruptured in the spleen and then they're eaten by the macrophages there, resulting into anemia. And finally, we have the aplastic anemia. In aplastic anemia, there is basically damage to the stem cells from which the red blood cells are to be produced. Now, if the stem cells are damaged, then the bone marrow cannot produce the red blood cells. Now, what are the causes of damage of stem cells? Now, conditions like SLE, systemic lupus erythematosus, or radiations or chemotherapy in the case of different cancers can result into aplasia. Even the insecticides used to kill insects in the farming, and even the substances like benzene, can cause damage to stem cells. So SLE, radiation, chemotherapy, insecticides, and benzene can damage stem cells, leading to aplastic anemia. So what is the treatment of aplastic anemia? Number one is we should stop the insult which is causing the aplastic anemia. And then we can give blood transfusions to counter the decrease in the production. Okay. Now, if we don't have any cause or we don't know the cause, then that is known as idiopathic aplastic anemia. Okay, so if the cause is unknown, then that is known as idiopathic aplastic anemia. So this is it. We have discussed basically the morphological classification of anemias and few other points regarding the anemias. Now we are going to discuss the effects of anemia on the circulatory system. That how exactly the anemia will affect the circulation. Now let's discuss it. Now, anemia means there are decreased number of RBCs. Now, whenever there are decreased number of RBCs in the blood, it means there is decreased internal friction when the blood is flowing. When we have more RBCs in the blood, then we have more friction between those greater number of RBCs. And when the count of the RBC is low, as in this case of anemia, then there will be decreased internal friction between the flowing blood. Now, as there is decreased friction, this will decrease the viscosity of the blood. Okay? So, and which will become 1.5 times of the water. Now, due to the anemia, due to the less cells, and due to the decreased friction, the viscosity of the blood is decreased. Now, if something has lower viscosity, then it means it is easy to flow. So that means this decreased viscosity will lead to increase in ease of blood flow to the tissues. Now, less cells, less friction, the blood can flow easily to the tissues. And whenever the blood can flow easily to the tissues, it will result into increased cardiac output. Whenever there is increased blood flow to the tissue, there is increased ease of flow of blood, which means increased blood flow to the tissues. For that increased blood flow to the tissues, we need increased cardiac output. That's why anemia is always linked to increased cardiac output. Okay? There's one more mechanism by which the anemia can lead to the increased cardiac output other than the decrease in viscosity, and that is whenever there are decreased in number of RBCs, it means there is decreased oxygen supply to the tissues of flowing blood. So there are decreased number of RBCs. RBCs have hemoglobin, and which carries oxygen to the tissues. When there are decreased in the number of RBCs, it means that less amount of oxygen is now being supplied to the tissues. Now, whenever there is less oxygen supplied to the tissues, it results into the dilation of peripheral blood vessels in those tissues. And whenever the blood vessels are dilated, it results into increased flow of blood to the tissues. And in order to keep the increased flow of blood to the tissues, cardiac output has to increase. So by this mechanism, cardiac output is also increased in anemia. So there are two mechanisms of increase in cardiac output because of the anemia. One is because of the decreased friction, and the second one is because of the vasodilation, because of decreased oxygen supply. Okay. Now, these two mechanisms are increasing the cardiac output in anemia. Now, interesting point is, as we have discussed earlier, that here there is decreased oxygen supply to the tissues because of anemia, but you can see the cardiac output is increased. So this decreased and oxygen carrying effect of anemia is counterbalanced by the increased cardiac output. Okay? Now, because of the anemia, the supply of oxygen to the tissues is decreased, but in anemia, there is increased cardiac output. So increased cardiac output counterbalances this decrease in oxygen supply to the tissues. So the oxygen are having a normal supply of oxygen in anemia, but this is at expense of the cardiac output. The heart is using its reserve. Okay, but the problem is when the person exercises, this anemic person exercises, now the cardiac reserve has already been utilized. Now the cardiac output cannot be further increased to such a level to provide the increased demand of oxygen of the exercising muscles. So that's why, and normally the oxygen is being supplied in the right quantity to the tissues, whereas because of the increased cardiac output, but in the case of exercise, that heart cannot cope with the increased demand because heart is already working way more than normal because of the already decreased oxygen supply. So the person feels extremely exhausted and extremely fatigued when the person exercises, when an anemic person exercises, and it might lead to cardiac failure. What is cardiac failure? The cardiac failure is basically that the heart is unable to pump the required amount of blood to the tissues, and the required amount of blood according to the oxygen demand. Okay, so basically the anemic person might go into the cardiac failure. Okay, so this is anemia, the types of the anemia and the effects of anemia on circulatory system. See you in next part. Hello everyone, this is Medical Gateway, and in this part we're going to study regarding polycythemia. So what is polycythemia? Polycythemia is basically increased in the number of red blood cells. So decrease in the number of red blood cells is anemia, whereas increase in the number of red blood cells is polycythemia. So polycythemia is of two types. One is secondary polycythemia due to some secondary cause, and then we have polycythemia vera or polycythemia rubra vera. Then we have, so secondary polycythemia. Polycythemia, increase in number of RBCs due to some secondary cause. So we have two causes here. One is high altitude and the other one is cardiac failure. So when a person goes to a place of high altitude, okay, the oxygen in the atmosphere is low. So when the person breathes in that low oxygened air, there is decreased oxygenation of the tissues. Now, the body will respond to this decreased oxygenation of the tissues by increasing the RBC production. As we have discussed in the erythropoietin, what was the main stimulus for the production of erythropoietin? The main stimulus was hypoxia. So whenever there is decreased oxygenation of the tissues due to any cause, due to the person is breathing a high altitude air, or because of the cardiac failure. In the cardiac failure, the heart is unable to pump the required amount of the blood to the tissues, or the heart cannot send the required amount of oxygen in the blood to the tissues. So that is cardiac failure. Heart is failing. The cardiac muscle is failing to send the blood supply to the tissues. There will be, so either it is because of secondary cause of high altitude, or it is because of some clinical reason like cardiac failure, the tissues are not being oxygenated well. And whenever the tissues are not being oxygenated well, this will result into increased RBC production. And this RBC production, whenever the number of the red blood cells are increased, there will be increased amount of friction in between those red blood cells, which will lead to increased viscosity. Okay? So there will be increased viscosity, and greater number of RBCs will lead to increased blood volume. So we have now blood having more blood cells, or more red blood cells, having greater viscosity, having a greater volume than normal. Okay. Now, if this secondary cause is physiological, then this is known as physiological polycythemia. So the cause in the high altitude is physiological. This is not some pathological cause, whereas cardiac failure is a pathological cause. So the secondary polycythemia because of high altitude is basically a physiological polycythemia. It's a physiological cause. It's the normal physiology working to provide more red blood cells. Okay, then we have polycythemia vera. Now in polycythemia vera, there is genetic defect in the hemocytoblastic cells. Now hemocytoblastic cells are the cells which are going to be differentiated into the red blood cells, white blood cells, and platelets. Now there is a genetic defect in these hemocytoblastic cells, and these hemocytoblastic cells have gone crazy. Okay, so they will multiply way more than the normal level of multiplication. Now, these blasts will keep on producing the red blood cells, the white blood cells, and the platelets. Okay, so we're just concerned with the red blood cells here because with reference to the polycythemia. So these increased amount of red blood cells will engorge the blood vessels. So vessels become engorged with these red blood cells. Okay, now, so as in the previous case, whenever there is increased production of red blood cells, there will be increased viscosity of the blood and there will be increased blood volume. So either it is secondary polycythemia because of it is physiological polycythemia, or due to some pathological cause, or because it's because of some genetic disorder like polycythemia vera. In all the causes, there will be increased blood viscosity, and there will be increased blood volume, and there will be increased number of red blood cells. Okay, now how these red blood cells are going to affect the circulation? Number one, there will be normal blood pressure in the case of polycythemia. But secondly, the most interesting point is regarding the venous return. So whenever the viscosity of the blood increases, it results into decreased venous return. Now the blood is flowing with a lot of friction, which makes the blood difficult to be returned to the heart. So this decreases the venous return. So amount of the blood returning to the heart in 1 minute decreases because the blood is flowing with greater viscosity. So increase in viscosity results into venous return. Now, but increase in blood volume will lead to increase in venous return. As you know, whenever the blood volume increases, the mean systemic filling pressure increases, which results into increased venous return, increased return of blood to the heart. Okay, so polycythemia was causing two things: one, viscosity, and the other one, increased blood volume. One, increased viscosity, and the other one is increased blood volume. So one thing is causing decrease in venous return, whereas the other thing is causing increase in venous return. The viscosity, increased viscosity, is decreasing the venous return, whereas increased blood volume is increasing the venous return. So by this, finally, the venous return, these two things are moving in opposite direction. So these two things counterbalance each other. So finally, there will be no increase or decrease in venous return, and as you know, the venous return is equal to cardiac output, so there will be no increase or decrease in cardiac output in polycythemia. Okay, so cardiac output will be normal. Why cardiac output will be normal? Because the venous return will be normal. And why the venous return will be normal? Because one factor is decreasing venous return and the other factor is increasing venous return. They both will counterbalance each other. So in the MCQs, if it comes, you will have to say that you'll have to take the option of normal polycythemia causes normal blood pressure and normal cardiac output. Okay, and normal venous return. Okay. Now, very interesting point with reference to the polycythemia. If I say a person is having polycythemia but along with the polycythemia, this person is having bluish or purplish discoloration of the skin, then you will think, why is this happening? The skin should turn only red. But what is happening, that whenever a viscosity of the blood is increased, there will be slow blood flow. There will be sluggish blood flow, especially in the skin subpapillary venous plexus. So underneath the skin, we have subpapillary venous plexus. Now, whenever the blood has a lot of RBCs, then due to increased friction, due to increased viscosity, the blood will flow slowly through those skin subpapillary venous plexus. And when the blood is flowing slowly, then a lot of the oxygen will move from the blood into the tissues. Due to the sluggish blood flow, tissues have more time to deoxygenate the blood. So, because of this increased viscosity in polycythemia, because of the sluggish blood flow in polycythemia, there will be increased deoxygenation of hemoglobin, especially in the skin subpapillary venous plexus. And you know, whenever the hemoglobin is deoxygenated, it will give a bluish discoloration, which is known as cyanosis. Therefore, because of increased deoxygenation, bluish discoloration of skin will appear, which is known as cyanosis of the skin. So that's why the polycythemia will give cyanosis. Okay, so this is regarding the polycythemia, its types, its effect on the circulatory system, and finally why the polycythemia gives a bluish discoloration of the skin, which is known as cyanosis. So this is it. See you in next chapter.